This conference will now be recorded.
So we have a few videos on aspergillosis. What are the different diseases caused by aspergillus? And then you can have a better idea about what sort of samples we'll be receiving.
Aspergillosis. Aspergillus species have a wide geographical distribution and are often found in decaying organic matter. Conidia are released into the environment and then are inhaled deep into the lung air spaces. In one of the classic forms of this disease, invasive pulmonary aspergillosis, the conidia are inhaled into the alveolar spaces, where the conidia germinate and become hyphae with invasion into pulmonary vessels. At this early stage, plain radiographs may not be revealing, but CT scan of the chest shows nodular lung lesions surrounded by a halo of low attenuation, reflecting hemorrhage around the fungal cell mass. Eventually, this lesion may cavitate with development of an air crescent sign, which is a favorable prognostic sign. Subsequently, with vascular invasion, the infection disseminates to liver, spleen, kidney, skin, and brain. This form of disease has an extremely high morbidity and mortality and requires aggressive surgical and medical therapy.
So this is a three-dimensional recreation of how the conidia will be growing on the surface. So anyone who is not muted their video, please mute yourself. Rema, call number 55.
Yes,
Rema?
madam. Yes,
Yeah,
madam.
please mute yourself, Amma. Yeah, thank you. Okay, so let us start on the PPTs. Okay, so wherever you cannot understand, you can just write in the chat box or just directly unmute yourself and say. So today's class is on Aspergillus. As we saw, the conidia growing on top of the vegetative hyphae. So this is an asexual mode of reproduction which we saw in the particular video. So let us go to the PPT. So basically, there are different types of fungi, like you might have discussed in your classes. So few are yeasts and few are molds. Okay, so you can see few are yeasts, few are molds, and some others are dimorphic fungi. So you might remember that dimorphic fungi have two types of morphologies. In certain temperatures, they will appear like molds, and in certain temperatures, they will appear like yeasts. So what are those different temperatures? So the body temperature that is about at 37 or at higher temperatures, these dimorphic fungi will appear as yeasts. Whereas in lower temperatures, that is at room temperature, usually where we grow the fungi, that is at 22 to 25 degrees Celsius, they appear as molds. So basically molds have different types of hyphae, as you might remember. Those are either aseptate hyphae or septate hyphae. So aseptate hyphae, you might remember, those are zygomycetes like Rhizopus and Mucor. It is easy to remember. How? Because these are bread molds. You might have remembered, like if you would leave pieces of bread or any other food items, if you leave them in a hot or warm moist place, so there you can see that some molds will be growing on them. So those are basically aseptate or sparsely septate. You might remember septa. Those are like divisions or walls in the middle of the hyphae. So septate hyphae, these have septate. So either they can be opportunistic fungi like Aspergillus or Fusarium, or they might be dermatophytes. Dermatophytes you might remember from your previous classes, like Trichophyton, Epidermophyton, Microsporon. So these are basically, they cause infection of the skin, hair and nails. That is the keratinized layer, they cause the infections. So these are different types of hyphae which we were discussing. That is, some will be aseptate, some will be septate. Aseptate means basically one or two septa may be there, so they are also known as sparsely septate. So some are yeasts, some are dimorphics, and some are molds. So dimorphic means at different temperatures they will appear in different morphologies like histoplasma, coccidioides, penicillium, etc. So at room temperature, that is 22 to 25 degrees Celsius, they will appear like molds. Whereas at the body temperature, that is 37 degrees, even if you put them in an incubator where you grow them at 37 degrees, they will grow as yeasts. Okay. So this is in short about what, just to recap, what you already remember about fungi. So basic shapes are either yeasts or hyphal cell types. So you might remember that maybe vegetative growths. For example, they will basically, yeasts will divide by budding. So candida, you might remember, you might have seen in your previous classes also, where we were showing you gram stain and all. So there you have seen candida cells, basically gram positive cells. So those will be very much larger than bacteria. So these will divide by budding, and sometimes what they will do is they will elongate. Those buds particularly, they will elongate and they will form something which is known as a pseudohypha. So that is, in the case of candida, they will grow as budding yeast-like cells or they will grow as pseudohypha. So then fungi have asexual development. For example, the conidia, like you saw in the video, the conidia will be growing on top of certain cells like vesicle-like structures. So they will basically bearing the conidia. So these conidia are basically spore-like structures. So they will be basically there in the air, and then these are the aerial mycelia. These will get tangled and these grow into the air, like you saw in the video. Whereas there will be some parts of the fungus which will grow into the particular substance on which it is growing. Okay. So then there is also sexual development in fungi. So there are different types of structures which are formed in sexual development. So those will take time. So because we have a applied class on Aspergillus, we are not discussing them today. So there are different types of fungi like dimorphic fungi. You can see at 25 degrees Celsius they grow as hyphae, whereas at 37 they grow as yeast cells. Okay. So these are budding yeast cells, whereas at 25 degrees, that is at room temperature, they grow as hyphae. So then you can see these are dimorphic fungi. So this is coccidioides. So there are different types of structures which are formed, and that is at yeast-like cells at the temperature 37 degrees, whereas at room temperature or at 25 they grow as hyphae. So these are particularly Candida is known as yeast-like cells. That is, they are not true yeasts. So they grow as budding yeast cells, and then they grow as pseudohyphae. That is the same budding. They will elongate and they will form something like which looks like a hypha, but those are not true hyphae. So that is why those are known as pseudohyphae. So this is a true yeast. Cryptococcus neoformans is a true yeast. So it also grows by budding. This has a large capsule. You might remember they have a large polysaccharide capsule and they divide by budding. Then there is Aspergillus, about which we will discuss today. So different types of conidia which will form. They have septate hyphae and these are hyaline hyphae. What do you mean by hyaline? Hyaline means glass-like or transparent. They do not have a color. There are different types of other hyphae also which are in pheohyphomycetes, in which there will be a typical color that is due to a melanin pigment. They will appear either brown or black. Whereas Aspergillus and all, they do not have a particular color to the hyphae. But why we see the color of the colonies, that is because of the different color of the conidia. So coming to Aspergillus species, there are more than 100 species. There are many species, out of which Aspergillus fumigatus is very common. And Aspergillus is found everywhere in the environment. The conidia are there in the air. They are found everywhere. And you might have inhaled, I might have inhaled, all of us might have inhaled. But because of our immunity, we are protected from the disease that is caused by Aspergillus. So they are found in soil, water, decomposing material. And there is a wide spectrum of Aspergillus associated disease. That is, very common is allergic bronchopulmonary aspergillosis, that is ABPA. It is now a well described form which is seen in cystic fibrosis, in old cavities where pulmonary TB cavities will be there. There can be fungus balls of Aspergillus. So these are not diseases caused by Aspergillus itself, but they are basically allergic diseases. So different roles are also there. So as we progress on in the class, we'll see. So here you can see this is a lactophenol cotton blue mound. You might remember that we have something which is known as lactophenol cotton blue. In that actually there is a blue color, and we prepare it as a wet mound. We put one drop of this lactophenol cotton blue, and from the colonies of whether it is Aspergillus or whether it is any other hyphal form, we just take a little bit of it on the tip of a sterile needle, and then we use another sterile needle to just basically just tweak it, or basically it is known as just we spread it on to the a particular place on the slide, and then we put a cover slip and see. So there what we can see is the different structures, that is how the conidia will appear. These are the phealides. These are the cells from which the conidia appear, and then there is a vesicle and the conidiophore, that is the bearing the conidia, so that is why it is known as a conidiophore. So all these things can be seen on a lactophenol cotton blue mound. So Aspergillus species belong to the kingdom fungi, to the phylum Ascomycota, and different families are there like Trichopomaceae. So the genus is Aspergillus, and it is a filamentous ubiquitous fungus which is found in nature. It is very commonly associated from the soil, plant debris, and even indoor air environments. So many species are there, and very few species are important to us. So that is why those only are taught to you. So many species are there like which produce aflatoxins. You might remember we have had a class on mycotoxins. So Aspergillus flavus produces aflatoxins. So if you take some, you might remember like peanuts. Suppose you take some peanuts which are particularly little bit of greenish color or have an off taste. So they might be having aflatoxins in them. So Aspergillus nidulans produces cutaneous aspergillosis, and oryzae. This is a helpful fungus. It is used to make soy sauce, sake. So those things are used to make these substances. Worldwide, it is found everywhere, and different species are there which are important to us. You have to remember only the top three, that is Aspergillus fumigatus, flavus, and niger. So Aspergillus terreus, lentulus, nidulans, many other species are also there. So this is Aspergillus fumigatus. Why I have kept this picture, so that you will just remember the color of the colonies. So you can remember flavus. Flavus always means yellow. So this is a sort of greenish yellow color. So the colonies are a little bit greenish yellow. Niger, as you know, it is always black. So Aspergillus niger is black in color. Fumigatus is basically a smoky green color. So like the fumes come out, so like that, it is a smoky green. It may not be that much apparent in this picture, but it is a smoky green color. This is the colony of Aspergillus terreus and nidulans. So these fungi, they have a very rapid growth, which is why they are very important culture contaminants in the laboratory. Suppose we want to grow some other fungi which grow rather slowly. So they, compared to Aspergillus, they have a very slower growth. So suppose you have not fumigated the laboratory, these Aspergillus organisms may grow as a rapid contaminant on the Sabroth's dextrose agar. You might remember that Sabroth's dextrose agar, those were there in tubes. So we have long, we have a slant of Sabroth's dextrose agar. There are different types in which Sabroth's dextrose agar, different types of antibiotics we can incorporate so that the bacterial so basic morphology is there is aerial hyphae, like we saw in the video. The aerial hyphae have conidia. These are there are long conidia, like you saw in the video. The conidiophores are long with terminal vesicles on which the phyalites, that is, they produce the conidia. They produce the biseptal chain of conidia. So different species variation there, like we saw, different conidia will have different arrangement of conidia will be different in different species. So we'll come to that. So here you can see the conidia, the phyalites are arranged all over the vesicle. So this is found especially in Aspergillus niger. So you can see all over the vesicle it is found. Whereas in some like fumigatus, you can see it is found only at the top part. Okay. So different arrangement of the phyalites and the conidia are found in different species. So in some, the structures are arranged all over. So this is a saprophytic fungus. There are many. They grow on decaying vegetables, on fatty media such as butter and ghee, on starchy media like bread and rice, on preserved foods such as jams and jellies, or like oranges and other fruits. So different on moist bread, like we discussed, mucor, rhizopus, penicillium. So they will also grow on moist bread. So different shades are there. These colors are different there. Different colors will be there. So conidial stage, that is the imperfect or the asexual stage, is found in this. Usually we see the asexual stage, that is the imperfect stage. So very few produce the perfect stage or the cleavage. These are the sexual forms. Okay. So 33 species are reported in India, and different species are used to make different food items also, like alcohol is prepared with oryzae, niger is used in the production of citric acid or other organic acids. Some species are used to yield antibiotics, and niger and oryzae yield a wide range of enzymes which are used for industrial fermentation. So they decay tobaccos, they spoil nuts and other foodstuffs. They grow on leather and fabrics and produce poisonous substances called mycotoxins also. Aspergillus produces like aspergillosis. Like in the human ear, they produce ophthomycosis. So this mycelium, you can see mycelium is actually a tangled structure of hyphae. So here you can see this is the asexual form, this is the sexual form, this is the pleistotecium, this is the perfect or the sexual form. So this archicarp is also a sexual form. This is the foot cell from which this particular conidiophore is arising. This is the asexual state. So this conidiophore is arising from here. It will bear the phyalides on top of that, and from the phyalides the conidia will be. So the mycelia structures, they are made up of interwoven mass, branched and septate hyphae. These hyphae are branched and form a mat on the substratum. So on whatever structure it is growing, like bread or any other substance, it will grow. So some of the hyphae lie superficially upon the substratum, and other penetrate deeply of the food and for the mycelia. So we are more interested in the asexual reproduction because that is why that is the thing which you will see under the microscope be showing you. So under a lactophenol cotton blue mound, you can see that there is a foot cell from which the conidiophore will be arising. Cell is a T-shaped cell. So this conidiophore will be arising vertically upwards, and then the phyalides or the stegmata will be there, and from there the conidia will be arising. So this is known as the vesicle. So the cells vigorously grow and become thick-walled. The thick-walled T-shaped cells are called the foot cells. Each T cell produces an erect branch called the conidiophores. So here you can see this is the wet mound. So these are the conidia. So round structures. So these are the conidia. This is Aspergillus niger. So you can very clearly. So this is the four, and these are the phyalides from which the conidia are arising. Okay. So the conidiophore length is around 1.5 is about 45 to 75 microns wide. The phyalides are biciliate phyalides. It is a typical structure. If you can remember or not. The color of the conidia is particularly black because we are seeing Aspergillus niger, and this is the diameter of the conidia, 4.5 to 5. It will vary a little bit among species, but usually among Aspergillus, you can differentiate with the color. So the length of conidiophores is around 5 millimeters, and the swells are the globostral structure called the vesicle. The lumen of the vesicle is continuous with the upper part of the conidiophores, and from the surface of the vesicle, tubular cells called the stegmata or the phyalides. The phyalides, the whole surface of the vesicle. This is in Aspergillus niger species. As I told you, it will be different in different species. So this is Aspergillus fumigatus. You can see the difference. So they are not covering the total thing. So different colors of the colonies you can see. This is Aspergillus fumigatus, a smoky green color. This is flavus. Always flavus means yellow. Niger. These three species we have. So fumigatus is the second most common as opportunistic fungal infection after Candida, and this species is the most common agent of aspergillosis in man and animals. It is a thermophilic species, that is, growth occurs at 40 degrees and beyond, and may be extremely angioinvasive, particularly in the immunocompromised patients. So these are all opportunistic fungi. That means whenever they get an opportunity, that is, the steroids, or is a transplant patient, or is on some sort of immunosuppressive drugs, is an HIV patient. So these organisms like Candida and Aspergillus, they will particularly you will find these organisms in those people. So this is Aspergillus niger. You might remember, black color, and the reason is because of the color of the conidia. You can see the color of the conidia. The hyphae will be typically, you can see this is the conidiophore. These are hyaline hyphae. So that means they have a glassy, transparent look. So this is the conidia. You can see the conidia. These are the round structures. Then they arise from the phyalides, and this is the vesicle. There is some other structure which you can find in some textbooks, that is known as the medulla. Here it is written in some other language. That is why it is the spelling is different. Otherwise, it is medulla. And then this is the conidiophore, and these are the hyphae. So conidia are basically asexual spores that grow on elaborate structures known as the conidiophores. These are usually stripping the conidia off the substrate for better dispersal and to avoid microscopic grazing animals. They often produce hundreds of thousands of conidia at a time, as you saw in the video. That is, these are long branching filaments that with other hyphae form the feeding thallus of a fungus called the mycelium. So they are intertwined or intertangled So a typical hypha consists of a tubular wall made up of chitin. Please remember. So chitins which surrounds, supports and protects the cells which compose a hyphae. So for most fungi, a cell within a hyphal filament is separated from other cells by internal cross walls called the septa. So that is what we were discussing in the initial part of the class. So these are hyphae. You can see these are typical septate hyaline hyphae. You cannot see any color. So this is a stained mound. So on that also you can see they have no color of their own. If it is pheohyphomycetes, melanin will be there, which is why they will be brown. They have no color of their own. You can see they are glass-like. So these are septate hyphae. You can very clearly see the septa. So very clearly you can see the septa. And these are the conidiophores with the conidia. So basically a fungus begins its existence as an independent biont, has some sort of propagule, most often a spore. We have seen about the conidia. So the fungus grows in a thread-like branching formation known as the hypha. The hypha grow and intertwine to form the mycelia, which becomes the body of the fungus. So different types of aspergillosis are there, like invasive aspergillosis can be acute or subacute. Acute is having a less than 1 month course, and subacute is acute or chronic necrotizing, lasts at least 1 to 3 months. Chronic aspergillosis lasts more than 3 months and can be divided into different types like chronic cavity pulmonary or aspergilloma of the lung or chronic fibrosis of the lung, chronic invasive sinusitis or maxillary or sinus aspergilloma. Aspergilloma, like a tuberculoma, is like a fungus ball. So pulmonary aspergillosis, which is the commonest, and then followed by CNS, sinonasal, osteomyelitis, endophthalmitis, endocarditis, renal abscess, and cutaneous. So why do you think pulmonary aspergillosis is the number 1 form? Naturally, because the conidia will be inhaled, like we saw in the video. So the fungal conidia are formed in abundance, which are aerosolized. They reach the lungs and then macrophages engulf and destroy conidia. But in the immunocompromised, that is the people who are having either leukemia, stem cell transplants, corticosteroids, macrophages which cannot contain the inoculum, that is the inoculum is very, the load of the inoculum is very high, the conidia will swell, they will germinate to hyphae in the cavities, and they will they will form sort of fungus balls also inside. So different as allergic forms are also there. That is IgE antibody is formed as the conidial antigen is there. So immediately asthma can be there. So allergic bronchopulmonary aspergillosis, in which the conidia germinate, the hyphae colonize the bronchial tree and the parenchyma is there. So aspergilloma, in which this is an extrapulmonary colonization. That is, they are inhaled on entering an existing cavity, like that formed by a tubercular cavity or something. They will go inside and then they will germinate to form the abundant hyphae in an abnormal space, like in tuberculosis, sarcoidosis, or emphysema. And patient may be asymptomatic or may have cough, dyspnea, hemoptysis, fatigue, weight loss. They may also invade the nasal sinuses, cornea, nail, ear, canal, etc. So here you can see in the air sacs also how this is actually in a hen, but the same thing happens in the humans. So this is a fungus ball. You can see totally hyphae also you can see very clearly. So this is made up of hyphae in the lungs. So asthma, recurrent chest infiltrates, eosinophilia, both type 1, type 3 hypersensitivity reactions can occur and permanent lung scarring can occur. Extrinsic allergic alveolitis, in which normal individuals with massive conidial exposure can be there. So here you can see Aspergillus fumigatus in lung tissue. So here you can see this is a staining which has been done. So the conidial head also you can see this is forming in an alveolus. The conidia also you can see, the hyphae also you can see. This is a histopathological staining which has been done, which is why you can see the color. Normally they will be colorless. Invasive aspergillosis, in which inhalation of the conidia will take place and then the conidia will germinate, and an acute pneumonic process. This is with mild necrosis to dissemination may or may not be there. And this invasive aspergillosis usually occurs in an immunocompromised patient. So suppose you get a case in which the patient is suffering with HIV or is some sort of immunocompromised patient in which the CD4 count may or may not have reduced. So suppose the CD4 count is less in AIDS patients, aspergillosis can be invasive, or stem cell allogenic hematopoietic transplant has taken place in a leukemia or a lymphoma patient, or the patient is on the corticosteroid therapy. So here you can see aspergilloma is found post-mortem in the lung of the child with leukemia. So fungus ball you can see very clearly occupying the cavity. So how to do lab diagnosis? Suppose you get a sample that is a sputum sample. So first of all, you have to do microscope. Naturally, you have to do a wet mount or a lactophenol cotton blue mount. So very clearly, you can see the conidia or the hyphae in specimens like exudates or like sputum. So the lung biopsy, respiratory tract, any specimen can be there. Bowel specimen can be there. Bronchoalveolar lavage. So you have to do a potassium hydroxide mount or a calcofluor stain or a histological mount, and then you can see the hyphae. So septate hyaline hyphae which branch dichotomously, that is you can see at about 45 degrees angle. There will be a 45 degrees angle of the branching septate hyphae. So then you can see the phthalates and the conidia also. Rate of growth, suppose you grow it on Sabroth's dextrose agar, the rate of growth is usually rapid. They mature within 3 days and some species are slower growing. That is the surface at first will be white. You have seen in the pictures also. They'll be white and then they can change to any shade of yellow, green, brown or even black depending on the species. The texture is velvety or cottony and the reverse is white, golden or brown. You can see the plates depending on the conidial colors. So fumigatus very clearly you can see smoky green color. So this is Niger very clearly. So microscopic morphology, that is the septate hyphae, you can see the unbranched conidiophore which arises from the foot cells. So we have to take from it, we have to make a lactophenol cotton blue mount. And then the vesicles are completely or partially covered with flask-shaped phthalates. The phthalates may develop directly on the vesicle, that is in the unicellular form or a bicellular form. If the material, that will be the bicellular form. And then the phthalates, we have seen the medulla, you might remember in the picture. So phthalates and medulla can be there. So the phthalates form a chain of round conidia about 2 to 5 micron in size. So you can see unicellular, that is the medulla is not there, no medulla. Directly on the phthalates, the conidia are growing. This is the bicellular phthalates, that is the medulla are there. First of all, on the vesicle, the medulla will be there. On top of that, the phthalates can be there. And on top of that, the conidia will be growing. So this is the foot cell from which the conidiophore is arising. And then there is the rounded vesicle on which the phthalates or the medulla will be there. On top of the phthalates will be there. And then the conidia will be basing on macroscopic morphology. You can see different pictures so that you can identify. Aspergillus fumigatus is velvety or powdery. At first it is white, then it turns dark greenish to grey with a narrow white border. The reverse is white to tan. So smoky green. Just remember fumigatus, fumes. You can remember fumes. So fumes to fumigatus. Okay. So smoky green color you can remember very easily. Once you come here, we'll be showing you the tubes. Then Aspergillus niger, it is woolly, and then the at first it is white, then it will be yellow, then it will turn black. The reverse is white to yellow. So niger you can very easily remember black. And then there is flavus, which is yellow to green in color, basically greenish yellow. Remember greenish yellow. Flavus always means yellow. Okay, flavy means yellow. So Aspergillus terreus is another species. I just want you to remember these. So So on microscopic morphology, you can remember that there are the conidiophores, then there are the phyalites, then these are the diagrams. So basically you can remember that fumigatus will basically, the phyalites will be growing in parallel to axis of the conidiophore, and it will not cover the entire surface of the vesicle. Whereas this in Niger and in flavors, they will cover entirely, the entire vesicle will be covered and a radiate head will be formed. So fumigators, basically if you can differentiate based on the color, you can differentiate between Niger and. So to conclude, there are many important species, out of which you have to remember only three, that is Niger, flavus and fumigator. In infectious as well as allergic diseases, there can be invasive disease, especially in the immunocompromised, which is why this is an opportunistic fungus. And based on the color of the cultures, that is the conidia will give you the culture different colors, and then arrangement on the lactophenol cotton bloom mounds, you can differentiate between the species. And basically these are laboratory contaminants also, which is why we have to be careful with our cultures. Okay. Thank you so much. We'll see a short video, after which if there are any questions or any other discussions we can do.
Aspergillosis is a disease caused by a fungus or mold called Aspergillus. The fungus is very common in both indoors, and most people breathe in the spores of the fungus every day without being affected. But some people get the disease. It usually occurs in people with lung diseases or weakened immune systems.
There are different kinds of aspergillosis. One kind is allergic bronchopulmonary aspergillosis, also called ABPA. Symptoms of ABPA include wheezing and coughing. ABPA can affect healthy people, but it is most common in people with asthma or cystic fibrosis. Another kind is invasive aspergillosis, which damages tissues in the body. It usually affects the lungs. Sometimes it can also cause infection in other organs and spread throughout the body. It affects people who have immune system problems, such as people who transplant, are taking high doses of steroids, or getting chemotherapy for some cancers.
Your doctor might do a variety of tests to make the diagnosis, including a chest X-ray, CT scan of the lungs, and an examination of tissues for signs of the fungus. Treatment is with antifungal drugs. If you have a burn, you may also take steroids.
So once we have discussed in the class about the conidia, the phyalites, the medullae, you can understand this video better, which is why I'm playing it again, so that you will remember it. You can see very clearly the top of the phyalites. Very clearly. To the rising. It's a three-dimensional creation, so you can understand it better. You can see the aerial and the vegetative hyphae. the mycelium. In total, the hyphae are very much intertangled. The conidia in the air, you can see they can be there anywhere in indoors air also, which is why we fumigate our laboratories and all. Okay. Thank you for joining us today. I hope that you and your parents are all well. Take care.